Mechanism of enzyme action. Factors that affect the rate of enzyme action and inhibition of enzymes, their inhibitors and their types of inhibitors. So first of all, let's start with enzymes. We know that enzymes are globular proteins, and proteins are always composed of amino acids. They are biologically active compounds that act as catalysts, and they increase the rate of reaction by decreasing the activation energy. They are very specific. Each substrate has its specific enzyme, and their presence doesn't affect the properties or the nature of the products that are formed during the enzyme reaction. So we will discuss some important terms here like activation energy, that is the minimum amount of energy required to start any enzymatic reaction. Now what is cofactor? The non-protein part of enzyme is called its cofactor, and this cofactor is required for the proper functioning of that enzyme. Examples include magnesium, copper, zinc, etc. Activator, the detachable cofactor, the cofactor that can be detached from the enzyme. If it is an inorganic ion, then it is called as activator of the enzyme. Now, if the non-protein part of enzyme is covalently or permanently attached to the enzyme, this is called prosthetic group. P for prosthetic, P for permanent. Coenzyme, if non-protein part is loosely attached to the enzyme, then this will be called as coenzyme. And as it is loosely attached, it can be detached and can be used again and again. Apoenzyme is an enzyme with its protein part removed, or we can say the non-protein part of enzyme is called as apoenzyme. And when protein and non-protein part both are present, then we will call this as a holoenzyme. Okay, now we will discuss about the mechanism of enzyme action. Okay, so we know that enzyme has its specific substrate, or substrate has its specific enzyme. When both of these combine, they form an intermediate compound which is called as enzyme substrate complex. This is a reversible reaction. This enzyme substrate complex can be converted back into enzyme and substrate as well. Now this enzyme substrate complex is then converted into enzyme, that is the same enzyme that was used, and the substrate is converted into the products. And this enzyme, as it is removed from the enzyme substrate complex, so we will use the term enzyme turnover here. This enzyme has two types of sites. One is active site and the other is allosteric site. The active site is that site where the substrate will be attached, and allosteric site is other site than active site. Now active site has two further subsites, the binding site and the catalytic site. Binding site is involved in recognition and binding of the substrate, while the catalytic site has catalysis activity, or we can say that this catalytic site converts the substrate into its products. Okay, now enzyme action can be described with two types of models, lock and key model and induced fit model. First of all, we will discuss about lock and key model that was proposed by Emil Fischer in 1890. This lock and key model states that only a specific enzyme can convert its specific substrate into products, and the active site of enzyme is very rigid and there is no change in its active site during the reaction. But according to induced fit model that was proposed by Koshland in 1959, this model states that substrate change the structure of the active site of an enzyme. Means the active site is a flexible structure according to induced fit model, and when there is change in the structure of enzyme, then this enzyme will perform its proper function and will convert the substrate into the product. Now we will discuss the factors that affect rate of enzyme action. First of all, about enzyme concentration. When there is more enzyme, there will be the faster rate of reaction. More products will be formed, more substrate will be converted into the products. But what is the condition here? That this will only happen when substrate is unlimited. If we have unlimited substrate, then enzyme is concentration is directly related to the reaction rate. Now substrate concentration. If there is low substrate concentration, then the increase in substrate concentration will result in increase of rate of reaction. What about temperature? If there is increase in temperature, obviously reaction rate will increase because temperature will increase the kinetic energy, and as a result the rate of reaction will increase. Enzyme will convert more substrate into the products. Now we have a term optimum temperature. What is optimum temperature? This is that temperature at which the enzyme has maximum activity, and for human body enzymes 37 degrees centigrade is that temperature at which enzymes act at their maximum level, and it is called the optimum temperature for those enzymes. The rate of reaction doubles for each 10-degree rise in temperature. If we increase the temperature by 10 degrees centigrade, then the rate of reaction will be double as compared to that it was before that rise. So there was a condition that if we will increase the temperature too much, this will result in enzyme denaturation. Enzymes will not work properly. The fourth factor is pH value. We have an optimum pH for each of the enzyme. Each enzyme has a specific pH at which it acts most effectively, and this is called its optimum pH. But if there is change in pH, this will result in amino acid hybridization. These amino acids are present at the active site, and as a result, the activity of enzyme will be reduced or blocked. And if there is extreme change in pH, then the bonds between the amino acids and enzymes will break, and enzymes will get denatured. They will not perform their function. Now, our last topic is enzyme inhibition. What are inhibitors? Inhibitors are the chemical substances, and they react with the enzyme at the place of substrate, and they will block the active site of the enzyme. As a result, substrate will not be converted into the products. Inhibitor examples include cyanide, poison, antimetabolites, drugs, etc. Now, we have two types of inhibitors, reversible inhibitors and irreversible inhibitors. Reversible inhibitors are those inhibitors that can be reversed. They form weak linkages with the enzyme's active site, and their effect can be neutralized or it can be reversed by increasing the substrate concentration. Now, reversible inhibitors are of two types, competitive and non-competitive. The competitive inhibitors are those that will bind at the active site of the enzyme where the substrate binds. But non-competitive inhibitors are those that will bind at site other than active site, or we can say that they bind at allosteric site or regulatory site. Now the competitive enzyme inhibitor example includes malonic acid, that is an competitive inhibitor of fumaric acid. Now, irreversible inhibitors will bind at active site. They will form permanent or covalent bonds, and they will destroy the globular structure, and their effect cannot be reversed. That was all. Good luck.